Today we talk about stem cells and related subjects. This is a major portion in CSIR UGC syllabus. This is the current topic, therefore it is more important in the examination point of view. Okay. So the stem cells, what are stem cells? That we have already actually you might be aware of what are stem cells is all about. However, I will start with this one. The egg is when it is fused with the sperm, this becomes the zygote. The zygote is the principal starting point for the whole embryo to develop or the whole fetus to develop. So this particular embryo, which can able to give rise to a complete embryo which is viable and also complete embryo and which is viable, also it produces all the organs that is actually inclusive of the organs present in all the three germ layers, or the organs or tissues present in the three germ layers. If it gives all these things, that means this zygote has the potential to do this. So this particular property is called totipotency. What is totipotency? The ability to produce the cells, tissues and organs of all three germ layers, first point. Second, to a viable complete embryo. If that definition is clear, it is called totipotency. So a zygote is a totipotent cell, meaning it can able to give rise to anything. Beyond zygote, what it happens here is that the zygote is now divides, say for example 2 divisions, 4 embryos. Now if you disaggregate the cells, otherwise you just remove the cells, then each one may form a complete embryo, but not beyond 4 cells. So that means a zygote loses its totipotency if it goes beyond 1 or 2 divisions. And now each cell, if it suppose it loses its totipotency, that means only, what do you mean by losses in totipotency? The one is that it can produce any organ or tissues or cells of the 3 germ layers, but it cannot produce viable embryo. This is the first thing it loses. So now the cells are said to be pluripotent. So what do you mean by pluripotency? The ability to form tissues, organs and cells of the 3 germ layers. 3 germ layers, but it cannot able to form a viable embryo. This is called pluripotency or pluripotent ability. So from the zygote, these are all stem cells. What do you mean by stem cells? The stem cells meaning is that it can able to produce different types of cells, meaning that the stem means it's like a stem. Okay. So now after certain times the zygote is going to divide and it can able to form a blastula. A blastula is having 2 layers with a blastocele, and there are only 2 layers of cells. Then it undergoes gastrulation. Now it forms 3 layers of cells. So there will be gastrulation, and this is the remnant of the blastocele. This is the gastrocele. Gastrocele is none other than the gastrulation inpushings, and there are how many layers now? There are 3 layers: ectoderm, endoderm and mesoderm. Sorry, this will be the mesoderm and the inner one will be the endoderm. So now you get into a gastrula where there are 3 germ layers are present. So that means ecto, meso and endoderm. Now from that each layer the organs now develops. From ectoderm we have the organs, that is it can able to form eye, it can able to form skin, it can able to form brain and other things. Even from ectoderm neural crust cells also forms in later phase. The neural crust cells now it can able to form pigmented cells of the skin. Pigmented cells, what are they? Melanocytes. Melanocytes of the of course the skin as well, they are formed from the ectoderm. And if you look at this mesodermal layer, that is in between ectoderm and endoderm, you have those that are present in our body from skin to the gut. What are the things in between? We have bones, we have tissue, we have bones, we have muscles, we have heart, we have blood, that is blood, vascular system. So these are all formed from the endoderm, mesoderm. Let us go into the endodermal one. So the one that is present inside is in none other than gut or alimentary canal. So the alimentary canal is formed by the endodermal layer along with those outpushings of the endoderm. For example, you have liver, that is formed by the endodermal outpushings, and also you have pancreas, that is formed from the endodermal outpushings. As a single unit it will come, pancreas and liver together it will come. So you have so these are the things you have to learn. So from gastrulation, from gastrula the fetus develops and the rudimentary organs, small organs, rudimentary organs are formed and then they increase in size. Okay, now what I would like to suggest here is that, so the ectodermal cells that are formed from the gastrula confined itself to form only a certain groups of organs and excludes others. This is a process of, one of the process of called commitment. The cell commits. It's called commitment. The commitment to develop into ectodermal organs. Similarly, the mesoderm also commits itself to form this one, mesodermal organs. Similar the case with the endoderm. So this primary commitment is called germ layer determination. It's called germ layer determination. The germ layer determination is the one that each germ layer determines what it wanted to become. So if suppose an ectodermal cell is forming an endodermal organ, or endodermal organ forming the mesodermal organ, suppose if the cell is doing it, this is called, that is called crossing the layer, that is crossing the germ layer, crossing the germ layer determination. Meaning a cell is crossing the germ layer determination that it has already set in. So every cell that is present in ectoderm, endoderm and mesoderm, though it is actually wanted to become a skin or this one, if you give proper condition, they can able to form any other type of the organs of the germ layers. So this is that is called what we call there is crossing the germ layer determination. Every stem cell can able to form this type of organ things. So this is, you have to learn this one. So what do you here, what does it mean by stem cell here? A stem cell is an undifferentiated cell, meaning, what do you mean by undifferentiated cell? The cell is not doing a particular function, a cell that is not doing a particular function. Why? An ectodermal cell here is not doing any particular function, though it wanted to do a bone or a skin or brain or whatever. It's not doing at present a particular function. An undifferentiated cell so capable of differentiating into any type of tissue, any type of cell or tissue. So this is undifferentiated. Second point, a stem cell has the ability of pluripotency. Pluripotency here meaning is that the cell is capable of forming any type of cell or tissue or organ, and it can able to cross the germ layer barrier. Germ layer barrier. What is a germ layer barrier? This is the germ layer barrier. That means a stem cell can, if you isolate from the ectodermal layer, but it can able to form mesoderm and endoderm, provided that condition is given to them. You understand? It can able to have a pluripotency. Third, transdifferentiation. Transdifferentiation. A cell, a stem cell, which you take it from the ectodermal layer, and it can form a cell of the mesodermal or endodermal. A cell which is taken from an ectodermal layer, which is destined to become suppose a neuron, can also form a skin. This is also a form of transdifferentiation. So you have a stem cell, these are called stem cell properties. Undifferentiated cell, pluripotency, transdifferentiation. Fourth, it can able to form lineages. Lineages means lines. What do you mean by line? For example, you take from mesodermal layer, you form the hematopoietic stem cell. Hematopoietic stem cell is the one that forms the blood cells. Okay. From this hematopoietic stem cell, there are two lines forms. One is myeloid one, another one is lymphoid one. Okay. See, the lines are going here. So here it can able to form both myeloid and lymphoid, but here it can able to form only myeloid one. The myeloid one further goes into macrophages, otherwise monocytes. Okay, monocytes. Lymphoid ones can able to go for T cells or B cells. Okay, T cells further divides into Th1 cells, Th2 cells, and further divisions, or CD4 positive cells, CD8 positive cells, NK cells, and like this. B cells of course forms the other things like plasma cells. And monocytes, you have megakaryocytes, and it becomes of course platelets, otherwise, and also myeloid granulocytes. Now granulocytes, you have example of granulocytes: neutrophil, basophil, and eosinophil. So from one stem cell there are lines going on. So of course a stem cell, now only one stem cell now divides into a group of stem cells, and certain groups forms one line, certain groups forms another line. This group of course further divides into lines. So this is called lineages. A stem cell can initiate and progress in lineages, so that ultimately a differentiated cell will be ensured. A differentiated cell will be ensured at the end of it. You got it clear about? So this is called lineages. So each lineage, you can identify a hematopoietic stem cell with CD34 marker, CD34 positive. Again another thing, SCOP plus, so many other things that we will discuss later. But however, what I would like to suggest here is this CD34 marker is no longer exist in a macrophage, meaning while it is passing through the lines, some of the markers will go off and differentiated markers will come in. So this is so that we can identify the groups, and each one will form the colony. Myeloid colony, lymphoid colony, these are the colony, a group of cells. From the group of cells, another group of cells will come. So these are all colonies. So that's why you say colony stimulation factors are involved in myeloid. For example, myeloid granulocyte monocyte colony stimulation factor is involved in myeloid colony formation. So similar things will come in, that we will go into later on when we go for hematopoietic stem cells as such, and its further things. But now we confined ourselves to the stem cell definition and characteristics. Lineage formation is one of the things, and then the stem cells has the ability to grow, multiply indefinitely. Multiply indefinitely. What do you mean by multiplication? Since it starts from very few stem cells, the other cells are formed. This stem cell must have the great multiplication potential. It should have the great multiplication potential. So if your cell wanted to divide for a longer time, what is the first thing you require is, you require the DNA to be replicated without any defects, and second, the DNA ends must be renewed or renewed at every division, because at every DNA every cell cycle a portion of the telomere will be cut off. So you require telomerase enzyme expression more. You require DNA repair enzymes level will be more. You understand? So again you get certain points here. So telomerase is in a high expression level in stem cells. DNA repair enzymes levels also will be expressed in a very high level. So what is the net point here is that since DNA repair enzymes levels are high, the stem cells, in contradiction to the general perspectives, the stem cell is in fact highly resistant to radiations. It is highly resistant to drugs. Example, there is a one is called ABCG2. ABCG2 is a drug transporter which is expressed in a very high level in stem cells, because it doesn't want it gets itself damaged due to even drugs or any exogenous materials, because when a stem cell is affected, the whole thing gets affected. So it wanted to protect itself to a maximum level, even that it expresses drug transporters, so that these are ATP cassette protein transporters, which requires ATP for efflux of drugs from the cells. So this is a cell, it has ATP cassette. This one, if you put some drug inside, they will be effluxed outside. So these are again the properties. High telomerase activity, DNA replication level is high, ABCG drug transporter thing also high, and also it expresses more amount of certain enzymes such as what we called as aldehyde dehydrogenase enzymes. Okay, so there are so many things, I mean, we can link off. These are also properties of stem cells. Okay, so what we have discussed now is, starting from the egg, we have come into a stage where there is a the fetus has formed. Now once it has the fetus has formed, you see, So now this becomes like the complete fetus. So in these things, before the organ forms, until the fetus forms, these are called fetal stem cells. Fetal stem cells. For example, a rudimentary of a liver. You take it as small cells. This is a fetal stem cell which can able to form the whole liver. So you can take it out. You take, for example, a rudimentary organ of the heart, the rudimentary tissues of the heart. You can take it out and culture it in the disc. It can form the heart tissue. So this is fetal stem cells. Once a child gives rise to birth, then what happens? Now in this process, every organ also have certain stem cells present inside. For example, you have a heart, you have heart stem cells. You have skin stem cells, or keratinocyte forming stem cells. And you have muscle forming stem cells, and you have of course the brain, and you have brain forming stem cell. Each has its own name, like ependymal stem cells as far as brain is concerned. Muscle, they are called oval cells. Skin stem cells, heart stem cells, like that. And also you have including stomach, you have a colon, rectum, any organ you can take. For example, eye has nimbal stem cells. In nimbal region, you have nimbal stem cells. So every organ of our body harbors a few stem cells which are required for the whole life of an individual. Hematopoietic stem cells is, of course, you know very well, which is useful for the formation of blood, which is present in the bone marrow. Okay, so every organ of our body now also harbors stem cells that is present, that is required for the whole life. What are the things that is required? One is that for the increase in size of the organ. Second, to replace worn out or damaged cells or tissues and other things. And third, in times of demand, it will be greatly increased. For example, anything in times of demand also it will increase, it will replicate. So this is the role of them. And if they are depleted, then you go for aging process. If they are depleted, you may end up in aging process. So this is another role where stem cells are bringing in. You understand? Clear about? So these are all you have to learn. And now I will come for so these stem cells are called adult stem cells. Why they are called adult stem cells? Because they are present in the adult organ. So this is a overview of what are stem cells. So from this, what are the things we have learned from this class is that the stem cells exist in embryonic stage. They are called embryonic stem cells. They are present in adult stage. They are called adult stem cells. They are present in fetus. They are called fetal stem cells. They are also present in umbilical cord, which is present there. So they are called umbilical stem cells. And they are also present in placenta. So they are called placental stem cells. Similarly, wherever it is present, it has its own name. And so, however, all stem cells has similar property. All stem cells have similar property, except few things. One may be totipotent, one may be pluripotent, one may be multipotent, and this is the difference. Totipotent, there is only one is zygote plus one or two divisions. Now pluripotent, all those of fetal stem cells are pluripotent cells, even adult stem cells also pluripotent. Multipotent, again all adult stem cells are multipotent, though they have the ability to become pluripotent. For example, a multipotent stem cell of hematopoietic stem cell. Why it is called multipotent? Because it forms a restricted tissue types such as blood cells. It is not going to form other things. Blood cells plus vasculature. Okay, or vasculature means endothelial cells. Endothelial cells. Now, since it is forming a restricted things, they are called multipotent. Now, they can become pluripotent. What do you mean by pluripotent now? So, HSC is a mesodermal stem cell, because blood and other things forms from mesoderm. They can able to form neuronal tissue, provided you give certain condition. For example, you give beta mercaptoethanol. You subject to beta mercaptoethanol, it may develop into neuronal cells or neuronal cells. This is called transdifferentiation, because it is crossing transdifferentiation. It is also crossing the germline barrier. Germline barrier. Why? This is a mesoderm, it crosses to become ectodermal type. So, this is also germline, crossing the germline barrier. So, this is some of the things you can bring about as far as stem cells are concerned. Also, as I talked about the adult stem cells, they are for the rejuvenation or for the growth of the organ, and it has its own types. It has its own types. They also can able to cross germline barrier. Able to cross germline barrier. So, and also the characteristics, one of the things I talked about, lineages and differentiation. Now, we now come into stem cell cycle. How the stem cell is dividing, or niche, and also we go for the niche. Now, we go into niches of the stem cells. What is niche? Stem cell niche. The niche is none other than the place where a stem cell resides. Stem cell resides. First point. Second, a place from where it functions. From where it functions. So, it is both a functional definition as well as a structural definition. Otherwise, it is not only a place it stays, but it also how it is a place where it functions from. So, it is a both functional and structural in nature, that is called as a niche. It is equivalent to for example, in ecology, a niche will be there. Similar definition is here. Okay. So, what happens? A stem cell, I told, is present in the adult organ. So, in the sense, if it is present in the adult organ, then it is where it present and how it functions and how it receives the signal from outside, that when it has to divide, when it should not divide. So, these are all the functions attributed to the niche. Okay. That means a niche of stem cell composed of few things. One is the cells that surround the stem cell, the factors that are secreted by the cells surrounding the stem cell. The third, the extracellular matrix around the stem cell. So, three components are present that constitutes the niche. The cells that surround the stem cell, the factors secreted by the stem cells, the ECM accompanying the stem cell. These three. What they does is they instruct the stem cell to divide or not to divide. It instructs the stem cell that you are a stem cell. You are a stem cell. It instructs the stem cell you are a stem cell. it tells the stem cell to divide or not to divide. Okay, in such a cases, you destroy a niche or you take the stem cell out of the niche. What it will do? First thing, it doesn't know what to do, divide to not to divide. So ultimately it progresses to division, because it is a stem cell. A stem cell property is to multiply, so it will divide. Second, it doesn't know whether it doesn't receive the signal to stay as a stem cell or not to stay as a stem cell. In that such situation, a stem cell goes for differentiation, meaning a little bit division and further differentiate into any type of cell that it wanted to become depending upon the condition. So otherwise it is forever lost. It's forever lost. If you take a stem cell out of the niche, it is forever, it is lost, meaning it divides and differentiates to something or something like this. You understand? So this is the importance of the niche. So the niche varies from different things. For example, hematopoietic stem cell niche is different from what is present in the stomach or of course the colon of the this one. It is different from the gonads. So every stem cell that resides here will receive different signals according to its function and structure. You understand? Clear about? So what is the adult and other instruction of the niche? Now we go for the hematopoietic stem cell niche first, then we can proceed with this. Okay. So hematopoietic stem cell niche. What is hematopoietic stem cell niche and how it is dividing, and other things we will look into it, which is also a part of your syllabus there. So now the hematopoietic stem cell niche, this is the bone, this is the bone of this one. So this is the bone, and there are vascular elements are present in between. That is, vascular elements means there is a blood will flow into this one inside the things. So this is endosteal niche. Endosteal niche. Okay, this is vascular niche. This is called vascular niche. Why it is called endosteal niche? It is the one that is very close to the bone. This is a endosteal niche and this is a vascular niche. Actually the stem cell is attached here. The stem cell is attached to a spindle shaped osteoblast, that is CD45 negative osteoblast. CD45 negative osteoblasts, and then there is a stem cell. This is the hematopoietic stem cell that is attached here. Okay, so how they does this? There are interaction. Now there is a cellular ones. There are a lot of stromal cells here. They are stromal cells, and this is vasculature. Here the oxygen level is very high here, very low here, oxygen level. And so what happens? So what are one of the property? In the low oxygen tension, this stem cell exists in this particular thing. And also fibroblast growth factors. Growth factors also is less here. So low growth factor level FGF4, that is FGF, fibroblast growth factor 4. So growth factor level is less, oxygen tension is less. Under this condition, this is the osteo, that is endosteal niche. So this is one of the characteristics. Now the cells and what are the cells we have here? The cellular components are osteoblast, and second thing stromal cells, which is surrounding this one. Okay, the factors that are secreted by them, and also the interaction between cell to cell, that is this cell to the stem cell, and then what are the factors secreted by the cell is also matters. And third, ECM. The ECM, extracellular matrix, holds that is the growth factors such as c-KIT. such as c-KIT. That is c-KIT, c-KIT. Stem cell factors, it has stem cell factors, it holds. So all these things are present. So now how it does, we will see here. That is, I'll erase this one. Now you have the spindle, that is a osteoblast. You have the connections here. There is one first connection is notch and delta connection, okay, and you have other connections like integrins and others, and then you have the receptors such as c-KIT receptors, and also you have WNT receptors, you have fibroblast growth factor receptors, and all these receptors are there. So what happens here is, that is, these are the stromal cells. I'll just give the brief idea. That is, so long as the notch and delta connection is there, the cell receives a signal that it is a stem cell. Okay. Other signals, WNT, c-KIT and other things, goes for multiplication. Also there are integrin signals that attaches the cell to the osteoblast. Okay. So what happens, unless this connection is severed, the cell will remain attached to the osteoblast and remain a stem cell. Suppose a cell is still attached and divides, it receives a division signal, it divides like this. So this cell and this cell still attached here, so this forms one stem cell into two stem cells. This is actually a symmetric division. It's called as a symmetric division because a stem cell gives us two stem cells. But if a cell receives a signal and divides into this way, then what happens? One stem cell remains attached to the osteoblast, whereas the other stem cell is given for further divisions and multiplication, which is not attaching to the osteoblast. This is called asymmetric division. Asymmetric division, where one cell is passed for division and multiplication and for differentiation. So what happens? This is the endosteal niche and this is the vascular niche. Okay, in the vascular niche, oxygen level is more, growth factors level is more, and everything is more. Here the cell divides and differentiates and then enter into the bloodstream. So here, in this asymmetric division, a stem cell, one stem cell gives rise to two cells, one is passed for differentiation, but still that one stem cell calculation still exists. Otherwise, here they used to call it as self-renewability. The cell is self-renewed itself, though it has passed one of its progeny to division. So a stem cell always self-renew itself, otherwise they are inexhaustible. Theoretically they are inexhaustible. In certain cases of damage to the tissue, for example, there is other things like for example for mobility, if suppose you undergo myocardial infarction or you are end up with liver cirrhosis or the severe liver damage, not liver, severe liver damage, where the stem cell requirement is more for rejuvenation of these tissues. Like for example, in myocardial infarction, the heart is damaged. Now the heart to sense the signal, I am damaged, and it requires assistance from bone marrow. It wants assistance from stem cells derived from the bone marrow. It sends certain signals. One signal is SDF, called stromal derived factor. So any ischemic means the one that is devoid of oxygen, sends a signal to the bone marrow how to recruit the stem cells. Now the stromal derived factor, which is of high concentration here, comes to the stromal cells and induces the expression of granulocyte colony stimulation factor. The granulocyte colony stimulation factor now induces the MMP, matrix metalloproteases, which cleaves the connection between the stem cells and the osteoblast, releasing the stem cells into the circulation for the rejuvenation of the myocardial infarction or liver damage. So this axis is called SDF CXCR4